particularly the anatomy of the neuron. And today, we're going to cover neurophysiology. So this is how neurons actually work. And because this material is somewhat complicated, we divide it up so we're going to talk about resting potential. That's resting membrane potential, so that'll be topic first. And then we'll see how that resting potential is changed or changes to create graded potentials. Graded potentials can produce an action potential, so we'll talk about that. And then ultimately, the action potential triggers the events at the synapse, which we probably will not get to until next time. And then from there, we get into the information processing, or how the nervous system makes decisions. So before we can talk about potentials, first you have to understand what we mean. So a potential is just a difference. So, if you took the battery out of the flashlight and you looked at it in terms of where the charges are, what you'd find in that battery is that all the positive charges are on one end and all the negative charges are on another. So whenever we have a separation of charge, in other words, the positives are on one side and the negatives are on another, we have a potential. Now, the unit that we use for electrical potential is voltage. So a 9-volt battery has an electrical difference of 9 volts. So what that means is that 9 volts worth of positives are separated from negatives. So if we take a voltmeter and we put one end on the positive and one end on the negative, the reading we'll get is the electrical difference. So for a 9-volt battery that's fully charged, you should see 9. Okay? So that's voltage. Now what we have in the cell, because we have ions that are moving around and ions that exist in the cell, just like we have a separation of charge in a battery, we have a separation of charge across the membrane. And we call that the membrane potential. We could call it the membrane voltage, but that isn't what we call it. We call it a potential because, and we use that word potential, because anywhere there's a difference, there's energy for change. Why do we make batteries? Well, because batteries push electrons around, right? The reason those electrons move is because of that difference. So we call it a potential because it's a potential energy. In other words, it can be made to go into motion. And what we'll see is that in the membrane potential, the cell uses that potential for movement to create that action potential, to send a signal from one end of the cell all the way down to the other. So in order to understand this resting potential, we have to understand how the cell is a combination of things happening at the same time. All right, so a little reminder. Sodium and potassium are not equally distributed in the body. So extracellularly, so in the extracellular fluid, we have very high sodium levels, but we have very low potassium levels. Inside the cell, we have the opposite scenario. Sodium is very low, potassium is very high. So our diffusion gradients, or our concentration gradients, are in opposite directions. So remember, substances want to move from areas of high concentration to areas of low concentration, right? That's diffusion. We talked about that early on in the course. And you can't forget it because we're going to talk a lot more about it next semester. So diffusion is moving from an area of high concentration to low. So for sodium, the diffusion gradient is toward the inside of the cell. Sodium wants to move from the outside to the inside because of that difference in concentration. For potassium, it's the opposite. Potassium is high inside the cell, low outside. So here, the concentration gradient is outbound. It wants to leave the cell. So if we were to suddenly make the cell more permeable to potassium, let's say, potassium would go rushing out because it has this big concentration gradient. Same with sodium. If we were to suddenly open a sodium door on the cell membrane, the sodium would go rushing in. Now, what makes this important is both of these are positively charged particles. So as they move, they not only bring their chemical nature with them, you know, substances move down their concentration gradient, but they also bring their electrical nature too. So when we open that sodium door, the sodium's going to come rushing in, and it's going to carry a positive charge with it, you know, because it's positive. Now, at some point, it can no longer come in because the inside, well, let me back up. As the sodium comes rushing in with its positive charge, the inside of the cell becomes more and more positive, right? Because positive charge is coming inside. Well, we know that like charges repel, right? Positives repel positives, negatives repel negatives. So as more and more positives enter the cell through that sodium door, at some point the electrical repulsion will actually stop the flow of sodium. In other words, it can't follow its concentration gradient anymore because there's too much positivity for it to come in contact with, so it's being repelled. The same thing would happen with potassium. If we open the potassium door, potassium's going to rush out, and it's going to take positive charge along with it. So at some point, and if positive charge is leaving, it's like an increase in negativity inside the cell. And we're going to talk about some of these in pictures here in a minute, but this is sort of the overview. So as more potassium leaves, the inside of the cell gets more and more negative, right? Well, as that happens, negatives and positives attract. So at some point, the inside of the cell will be so negative that the potassium can't leave because it's being held in place electrically, even though it wants to move chemically. So this resting potential is kind of a balance between chemical motion and electrical motion. And because sodium and potassium have a positive charge, their movement is going to affect the membrane potential of the cell. All right, so let's look at that in some different ways here. Okay, I think we covered all of this. Ions. So the sodium and potassium difference is maintained by a pump called the sodium-potassium pump. Because the cell, it needs to have it be like this. It needs to have the sodium high outside and low inside, and the potassium high inside and low outside, in order to make these action potential things that we're going to talk about happen. The problem is the cell membrane is leaky. In other words, sodium is always leaking in, and potassium is always leaking out. So it's not perfectly sealed. So in order to account for that leak, there's a pump that pumps sodium out and potassium in. So we call that the sodium-potassium pump. And the direction goes like that. So sodium is pumped out, potassium is pumped in. Why? To maintain this difference. High outside, low inside, high inside, low outside. You will hear about the sodium-potassium pump in your futures because without it, cells die. So it's one of the more important proteins that we find on the cell membrane is this sodium-potassium pump. All right. Okay, so looking at this, so we're zoomed in here. Here's the plasma membrane, so cell membrane. In orange here are potassium ions. So you see that there's lots of those inside the cell, right? But not very many outside the cell. And then the blue circles are sodium. Lots of sodiums outside, not very many sodiums inside. And then we have channels. The channels are what make the cell membrane leaky. Potassium can always get out, so it's leaking out down its concentration gradient. And sodium is leaking in, again, down its concentration gradient. And then here in the middle is that sodium-potassium pump, pumping sodium out and pumping potassium in, but that takes ATP, so it requires energy to do that. Now, this pump is very, very efficient because it actually pumps more than one ion at a time. So it'll pump 3 sodiums out and 2 potassiums in. So all told, sodium is leaking in, potassium is leaking out, the pump is pumping sodium out and potassium in. When we go and actually measure the membrane potential, we get minus 70. So that number is worth remembering. So the resting membrane potential of most neurons is minus 70 millivolts. Now, what does that mean? Well, the number, the 70, that's the magnitude. How much electrical difference is there across the membrane? So what that means is that there are 70 charges, either negatives or positives, that have been separated. So the electrons have been separated from the protons. And then, let's see, so minus 70. And what we'll see is that resting potential is going to change as we affect this balance. So if we were to, say, open a bunch of sodium channels, sodium would come rushing in, and that would increase the membrane potential. We would go from minus 70 to plus 60. So what does the minus and plus mean? All that means is what kind of charge is on the inside of the membrane. So, normally we think of negative numbers as almost a kind of subtraction. Well, here what we're saying is there's a magnitude 70 difference, and the charge on the inside of the membrane is negative. Because what we're going to see is as action potentials happen, it flips. So instead of having negatives along the inside of the membrane, like you see there, we're going to have positives along there instead. So this minus 70 means there's a magnitude 70, and then the charge on the inside of the membrane is minus here. All right. Some other players in terms of the electrical activity. Out in the extracellular fluid, we have chloride. Chloride is a negatively charged ion. And then inside the cell, the negative balance is provided by proteins. Most proteins have a negative charge on their surface. So the reason that the cell is always in electrical balance is because there are always enough minuses around to balance out all the pluses. But the reason we get a membrane potential is they're not always in the same place. So the negatives and positives end up getting separated. All right. So one of the ways of, why minus 70? Where does that come from? And it comes from the balance of sodium leakage and potassium leakage. So one of the ways we can look at this whole membrane potential idea is by looking at some made-up scenarios. So let's say that we took the plasma membrane and we made it impermeable to sodium, but freely permeable to potassium. In other words, all the potassium doors are open, all the sodium doors are closed. So, given the concentration gradient, if we opened all the potassium doors, potassium's going to go rushing out, right? And if a positive leaves, that's going to leave a negative behind, right? So if we did that, what we would find is that the cell would stabilize at minus 90. So a magnitude 90 difference, and inside the cell membrane would be a negative charge. Now, why minus 90? That's the potential at which the electrical attraction of positive to negative stops the flow of potassium out of the cell. So it's an equilibrium. That's the point at which no more potassium leaves because the inside of the cell is so negative that it's being pulled on too much to actually move anymore. All right, so that's the potassium side of things. If we look at the opposite scenario, so we close all the potassium doors, but we open all the sodium doors. Well, what would happen? Sodium's going to come rushing in from high concentration to low, and as it does so, it's going to bring positive charge with it, because the sodium ion is a positive charge. So if we left that system alone, what we would find is instead of minus 70, we would have plus 66 as our membrane voltage. So very different number. Potassium channels minus 90, sodium channels plus 60. So how does that happen? Sodium comes rushing in and it brings its positive charge with it. Well, at some point there's going to be so much more positive inside the cell than there is outside, that the sodium's going to stop moving because it would have to go against the like charge. You know, positives repel. So the sodium movement is stopped by that electrical repulsion. So the reason I bring these two scenarios up is in the action potential, we're going to see the membrane potential go from -90 all the way up to +60 and then back. And the reason for that is because those are the sodium and potassium potentials. So sodium has a tendency to make the inside of the cell positive. Potassium has a tendency to make the inside of the cell negative. They're movements, in other words. The movement of potassium makes the inside of the cell negative, sodium positive. All right. Questions about any of that before we go on? So, membrane potential brought about by the movement of these ions. And I think you'll see it'll start to come together here as we put this in motion. All right. Oh, that's right. We got a movie first. All right. So this is about resting membrane potential. I think he's going to talk.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at -90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to -70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
And we talk about membrane potential when we talk about the cells that are the active cells, like muscle and nerve, because they're resting membrane potentials, their membrane potential changes. All cells in the body have a membrane potential, and different cells have different potentials, but -70 is the typical human resting membrane potential. But we don't really hear it much talked about when we get to like epithelial cells because they don't do anything with that membrane potential, but muscle and nerve does. Okay, so at -70, we are not at the equilibrium potential for either sodium or potassium. So that means that if the permeability of the membrane changes, either to sodium or to potassium, things are going to move because they're not at equilibrium. Now, by holding the membrane potential at minus 70, which is what the sodium-potassium pump does, it means that when potassium channels open or close, or sodium channels open or close, we're going to see a change in the membrane potential. And the way the body uses this is, just like sending signals through a wire, like Morse code, you can say on or off. Well, the neuron uses this membrane potential as a way to send a signal from one end to the other, as we'll see when we get to action potential propagation. All right. So there are three different kinds of sodium and potassium channels that can change rapidly. So some open or close in response to voltage, some to the presence of a chemical. These are neurotransmitters, so obviously those are going to be important. And then we also have mechanically triggered gates. So here at the top is a chemically gated channel. This will be typical for a neurotransmitter receptor. So here, extracellular, intracellular, the gate is closed, right? So stuff can't get through. Throw a little acetylcholine in these pockets right here, though, and this channel will open. And in this case, this is a sodium channel. So when the acetylcholine binds, the sodium channel opens, sodium comes rushing in. When sodium comes rushing in, it's going to affect this resting membrane potential, because the minus 90 is going to go up towards 0, because positive charge is going to come in and start to offset the negative charge that's present there. All right, so that's one form. We have voltage-gated channels. As the membrane potential in a particular area changes, we have proteins that can open or close their gates to allow ions through. So like here, the gate is closed, but if we can drive this membrane potential up to minus 60, we can pop this gate open. So this would be called depolarization. So going from minus 70 to minus 60 opens this door. Once it's opened, sodium comes rushing through, right, down its concentration gradient. And now we see the membrane potential go way up, because this positive charge is coming inside. It's going to change what you see on this meter. Now at plus 30, the other gate closes. So at minus 60, this gate opens, and at plus 30, this gate closes. Sodium channels have this second gate called the inactivation gate. So when the membrane potential in that region hits plus 30, basically the door is closed, but it's a different door, which is a detail that we're not going to get very much into in here, but just know that there's two gates on the sodium channels. And then the last kind of gated channels are mechanically gated channels. So, how do we sense touch? What touch does is it deforms the membrane. So we have these cells in our skin that, obviously have cell membranes like all the others. But if you deform one end of the membrane, you can actually open a channel allowing sodium ions in or potassium ions out. And that difference can actually trigger an action potential, which then brings information up to the brain. So these mechanically gated channels are found not only in the skin, but also in the hearing and balance systems found in the inner ear, which we'll talk about more later. But for this chapter, it's these top two, the neurotransmitter gates, also called chemically gated channels, and then the voltage-gated channels. These are what are responsible for actually generating that action potential. All right. So we have a resting membrane potential, and we know that it can change. If we open one kind of gate, we can make that minus 70 a plus 30. If we open the others, we can make it less or even more negative. So we can change that membrane potential. So just for the sake of common words, we use the word depolarization to talk about a change in membrane potential to make it more positive. All right, so if this is, here we go, we'll do it this way. If this is plus 60, and this is minus 90, and this is minus 70, okay? So this is depolarization, Okay? So making less negative, heading towards zero, becoming more positive, all those same things, in up direction. So depolarization is a change in membrane potential towards positivity, and then the other way is repolarization. So when we go from positive back to negative, that's repolarization. And then down here between minus 70 and minus 90, this direction is hyper. So this is hyperpolarization. Going, in other words, becoming more negative than it is at rest is hyperpolarization. So depolarization, repolarization, hyperpolarization. All right. Let's see. So typically, when sodium channels open, positive ion comes rushing in. So more positive inside means less negative, right? So sodium channels usually cause depolarization. And then repolarization and hyperpolarization are usually the result of potassium channels, or positive charge leaving the cell, leaving negative behind. All right. So a graded potential, also called a local potential, is a change in membrane potential in one small region of a cell. Okay. So, nerves or neurons connect to one another. So what we have here's a cell body, right? Here's the axon of one neuron. Here's the axon of another. And then this is the beginning of the axon of this cell. So we've got 3 cells here that are interacting. Cell 1, cell 2, and then our target cells, or cell 3. All right. So cell 1 has an action potential that travels down this axon all the way to the synapse. At the synapse, neurotransmitter is released. That causes some chemically gated sodium channels to open. So if we look just right here at this area of membrane on this cell, we would see that the membrane potential would change. Here it's at rest. That action potential comes down here. The neurotransmitter is released, and we get a little depolarization. Why? Because this neuron released a little acetylcholine that triggered the opening of some sodium channels. So some sodium channels open, positive comes rushing in, and we see a little depolarization. It becomes a little less negative. Now then it goes back to normal because the cell corrects for that. And then let's say from this cell, instead of acetylcholine, we get a different neurotransmitter that instead opens potassium channels. So when it fires, we'll get a little decrease in membrane potential, or a little hyperpolarization, because some potassium rushed out and dropped the membrane potential and then came back up. Now, the magic happens when we reach threshold. So for most neurons, when they reach, when the axon hillock, or the very first part of the axon, when it reaches minus 60 as a membrane potential, that is what triggers an action potential, which is a whole cell event. These graded potentials, they don't affect the whole cell. They only affect part of it. And what happens is, depending on how they stack up, so to speak, if you have a response here and here, let's say they were both depolarizing. So this caused some depolarization, and then the other one did the same thing. Well, now instead of having a change like this, you'd have a change like this, a bigger change, and that would trigger an action potential. So graded potentials are what eventually trigger the action potential, and that happens at the axon hillock. All right, so what is this thing? What's an action potential? The simplest way to describe it is it's a change in membrane potential that sweeps across the whole cell. And because of these voltage-gated channels, it's possible to do that. Now, action potentials have some rules. They are an all or none event. In other words, a neuron can't have half an action potential. It can either have one or not have one. And the determination as to whether it's going to fire or not is based on adding together those graded potentials. So if the cell is brought to threshold, in other words, if that first part of the axon, that axon hillock, if all the graded potentials in the cell add up to that area getting above minus 60, then the action potential is going to stream down the axon. All right. So the graded potentials working together, if they reach that threshold, minus 60, at that minus 60, we have voltage-gated channels that open. Sodium channels do. So when the membrane potential gets to minus 60, sodium channels open. Okay, what would happen? Well, if sodium channels open, sodium is high outside, low inside. So sodium would come rushing in. So here, this is at rest. Graded potentials bring this minus 70 to minus 60. At minus 60, the gate opens. Sodium comes rushing in, and what do we see in the meter? We see depolarization. We've gone from minus 60 to plus 10 simply because this sodium channel is open. So sodium comes rushing in. Now at plus 30, that sodium gate inactivates. So the second gate closes, and at about that same voltage, plus 30, now the potassium channel opens. So far it's just been sodium. So depolarization threshold, minus 60, gate opens, sodium rushes in, gets to plus 30, gate closes, gate opens. So while one gate closes, another gate opens. But potassium is going to move in the opposite direction because potassium is high inside and low outside. So once the sodium gate is closed and the potassium gate opens, potassium rushes out, and what do we see in the voltmeter? A drop back down to negative. Why? Because potassium is carrying positive charge out of the cell. Sodium is bringing it into the cell, so we see depolarization. Potassium is taking it out of the cell, so we see repolarization and then even hyperpolarization as we get to minus 90. At minus 90, that triggers the closure of the potassium gates. So the gate closes, and now nothing's moving, right? The sodium gate is closed, the potassium gate is closed, but so how do we get back to minus 70? That would be the job of the sodium-potassium pump again, to put everything back the way it was so that an action potential could happen again. So if we look at this in a graph instead of in a concept diagram like that, if we were to look at the membrane potential at one particular spot on a neuron, as an action potential passed, this is what we would see. So here's at rest, so it's sitting there at minus 70, not doing anything. And then at 1, this is bringing it to threshold. So these are those graded potentials. Might be from chemically gated channels, mechanically gated channels. Either one of those, if this region reaches its threshold at minus 60, then we see this big thing happen. All right, so at number 1, we have that graded depolarization, getting that cell to fire, its action potential, getting it to its threshold of minus 60. All right, at minus 60, the voltage-gated sodium channels open, so we see this dramatic increase in membrane potential. Why? Because positive ions are flooding into the cell. So the membrane potential is becoming less negative. It's depolarizing, heading up to that sodium equilibrium potential. So that's at number 2. At number 3, at plus 30, the sodium channels close and the potassium channels open. Potassium moves in the other direction. So positive ions are flooding out of the cell, so the membrane potential drops all the way down to approximately minus 90, which is the equilibrium potential for potassium. At that point, the potassium channels close, and now the cell is in its refractory period. In other words, its recovery period. A lot of ions have moved around, so a nerve can't fire again instantly. It has to put everything back the way it was before it can fire again. So one of the limitations on the speed at which neurons can fire is the physiology of the action potential. You know, it takes time for all those ions to get back into their normal place. All right, so let's look at an action potential. So essentially, this is driven by those voltage-gated channels. All right.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only, and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary: generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
All right. So we have this kind of multi-step process that triggers a pattern of change in the membrane potential. So when you see that graph like that, just remember that that is looking at just one part or one spot on the neuron cell membrane. Because what action potentials move, the idea here is to transmit information from the cell body all the way down the axon so that the next neuron in the chain can be affected. So this action potential, this pattern, is we see that pattern of change as we go all the way down. So like, let's say we had this big long axon, right? And we were going to study the membrane potential right there. So as an action potential moves down, we'll see, just like the waves of an ocean, you see it go up and then that wave moves. We see the same thing. So we'll see this pattern here, and then here, and then here, and then here, and then here as that membrane potential change sweeps down the axon. So just remember that this is in one spot, and you won't get confused when we get to the next spot. Yeah?
So none of this requires ATP, but is ATP necessary, like a secondary for all of this to actually happen?
Yes, because ATP powers the sodium-potassium pump,
Okay.
which makes this potassium and sodium difference happen.
Okay.
So essentially, that pump is creating all the energy for this motion.
Okay.
So good answer. But no, the closing and opening don't require any ATP. All right. Okay, so like I just said, the action potential, that pattern, is at just one spot. Now, the interesting thing is an action potential in one part of a cell will create an action potential in the neighboring part of that cell. So, and the reason for that is, the first stage of the action potential is the sodium channels open. Sodium channels open, sodium comes rushing in, and the cell membrane, the membrane potential goes from -70 up. Well, remember, the threshold for opening sodium channels is -60. So as the sodium comes in, so we'll look at like 3 areas. So 1, 2, and 3, right? So here's 1. It's been brought to threshold. So sodium has been rushing in. We're at a membrane potential of +30. Well, the sodium, it doesn't stay put. Sodium is an ion. It moves very freely in fluid. So here we have a negative area in segment 2. The membrane potential is -70, right? Which means that there's a whole bunch of negative charge on the inside of the membrane. Sodium is positive, so what's it going to do? Well, it's going to flow towards the areas of negativity, right? Because opposites attract. So here the sodium is cruising along down the inside of the membrane, and as it does so, it changes the membrane potential. So here we go from -70 to -60 because the sodium from the neighboring segment has made its way over. So this -60 does the same thing, triggers the opening of sodium channels. So now we get here. The membrane potential is +30. Sodium has rushed in, but the sodium is going to be drawn to the next segment, which is still at -70. So we get this is called continuous propagation. One region's sodium moves to the next region, the distal region, the next one down, and that movement triggers that change in membrane potential. So we get this flow of membrane potential change that goes all the way down the axon. So, we divide it into segments, but the truth is it's kind of continuous because you could make these segments smaller and smaller and you'd see the same pattern. So the sodium moves towards the next area, depolarizes the membrane, that opens sodium channels, which then does the same thing for the next segment, and so on, until we get all the way down to the end of the axon. Now that's slow. The problem with continuous propagation, it works just fine, but to depolarize all that membrane takes a lot of time. So the body has evolved a different mechanism for speeding this process up, and that's called saltatory propagation. And this is the reason why we have myelin. Remember, myelin insulates the axons of some neurons. The reason that myelin is there is because it speeds up this action potential propagation. But it doesn't change it. It's still the same process. It's just we can skip areas of membrane. So here, instead of having 3 segments that are right next to each other, we have 3 segments that are separated by areas of myelination. So we call these empty spots between the myelin areas. Those are the nodes of Ranvier, or just the propagation nodes. So here we've got +30. So sodium is rushing in because this was brought to threshold. And here again, that sodium doesn't stay put. It moves along the inside of the membrane. But because there's myelin covering this part, nothing can happen there. The inside of the axon is insulated from the outside. So we don't get any change here Instead, the sodium ions just flow to the next open spot, where then they do the same thing, and then we get the same thing again. So it's the same process as continuous propagation. It's just been spread out, and there's some distance put between the segments by those myelin nodes. So essentially, the reason this speeds it up is all that has to move in this area are the sodium ions. And sodium ions are very small, so they're very fast. So they can zip along the inside of this myelinated axon and get right to the next area where it'll trigger the next thing. So we talk about this as saltatory. Saltatory means leaping. And it's the membrane potential change leaps from one node to the next until it gets all the way down to the end. So this speeds up conduction. All right. And this will be our last one for today. We're not going to get to the questions, sorry. So some things about nerve fibers in general. The larger the nerve fiber, or the larger the axon, the faster the conduction. So some of the fastest nerves in the body are also the largest nerves. And then we have type A, type B, and type C fibers. Type A fibers, this is like, the high-speed fiber optic internet that has been invented. These are fast, fast, fast. So they're myelinated, they're large diameter, so they can send signals at up to 268 miles an hour. So that's very fast. The type B fibers are smaller, but are still myelinated. They can send at about 40 miles an hour. And then type C fibers are the oldest kind of neurons. They don't have myelin. They're very thin fibers, very slow. You can experience the difference. If you've ever stubbed your toe, and I'm sure we have all done that, the initial pain comes very quickly, right? You know about it immediately. That's type A fibers, coming all the way from your big toe, all the way up to the noodle in your head, in that short amount of time, so very fast. Now then what happens a couple minutes later when you start to feel the pain of it, right? The throb or the ache, those are type C fibers. It took them all that time to send that information up to the brain because they move so much slower than the type A fibers that told you you stubbed your toe in the first place. All right. Thank you.